Welcome to our webinar on the future of technology, biotechnology and mining, hosted by BRIMM, the Bradshaw Research Institute in Mining and Minerals at the University of British Columbia, and MICA, the Mining Innovation Commercialisation Accelerator, which is a national Canadian federal initiative to accelerate the introduction of new technology into mining. To start out with, UBC is located in Vancouver, and the Vancouver campus is on the traditional, ancestral and unceded territory of the Coast Salish peoples, within the territory of the Musqueam Nation, also the Squamish Nation and the Tsleil-Waututh Nation. We would like to acknowledge the land on which each of us is currently located, wherever you are. Unfortunately, we've been unable to set up the French interpretation function, but please post your questions for the panel in the Q&A channel and use the chat for discussions using the webinar. We will monitor the Q&A during the discussion, and we're running to a fairly tight time schedule, 15 minutes per speaker. So there won't be questions after each speaker, but there'll be a discussion time at the end. So without further ado, I would like to introduce you to Dr. Aria Hahn, who is the founder and also the CEO of bioinformatics company Koonkie. Over to you, Aria.
Hi, thank you. I'm just going to get screen sharing on here. I'll make this big. Yeah, thanks for the introduction, John. So my company is called Koonkie. It's a big elephant. It actually has a ton of history, and it goes actually nicely with microbes, which is really what we focus on. So microbes are small, and we're thinking big about them, which is partly where this elephant comes from. Koonkie elephants are also found in Assam, India, which one of our original co-founders was from there. And he would tell me these stories. He often talks in parables, and he would tell me these stories about these Koonkie elephants capturing, taming and managing these wild elephants and bringing them to domestication. And we really liked that as an analogy for what we're trying to do with big data. So even though microbes are absolutely tiny, you know, there's a lot of them. So I'm just going to put this into context with some of my favorite slides of all time. So one of the most universal feelings I think everyone can relate to it is you're out at night somewhere where you're outside of city lights. There's not a lot of light pollution, and you're looking up at the sky and you see all the stars, and you get that feeling of being really small, but at the same time, it's really pleasant. And so there are 10 to the 22 stars in the known universe. And it's a lot. But that's absolutely eclipsed by the number of microbes that are here on planet Earth. So this is an image taken by Ed DeLong, who works a lot in metagenomics, which is what we mostly focus on at Koonkie, which means we study microbial communities. They don't exist. Microbes don't really live on their own. And so there are just so many microbes. Sorry, somehow I got muted. There are so many microbes on the planet that it's absolutely this incredible feeling. And so if you get that feeling of smallness when you think about the night sky and the stars, you should get that feeling orders of magnitude more when you're just thinking about microbes. I like to show this slide because usually when people think about DNA, they think about Watson and Crick and this double helix and this structure. And yes, it's a double helix and it's a great symbol, but it's actually one of my least favorite, it's one of my least favorite ways to think about DNA. And so now you're probably thinking, this is a mining conference and all you're talking about is microbes and DNA, and I swear it's all going to come together. So microbes, this is kind of an analogy that I like to tell people is microbes don't live in isolation. And so often when I say I'm a microbiologist, people think microscopes, they think plates, they think E. coli. All those things are true, but I've never used a microscope in my life. And I don't grow microbes. And we don't grow them because most microbes, well over 90%, some people say 99%, 90 is probably a little bit more accurate, but 90% of microbes in the world, these little bacteria, things that you can't see that are absolutely everywhere on this planet, can't be grown in the lab to the best of our knowledge and technologies today. And so people often say, well, why? They're so small, like, why can't you grow them? And a good way to think about it is this. If there was a giant that was studying humans, and they knew something about you, they're like, yeah, eats a lot of corn, eats a lot of beef, breathes this nitrogen, oxygen atmosphere, and they scooped you up and they put you in a little box, had the right atmosphere, then they dump a bunch of corn and a cow in there, you're not gonna live that long. What's going to take care of your waste? How are you going to cook that cow? How are you going to butcher that cow? Like, what are you going to do with a live cow? Not a lot. And what are you going to do with, a couple kernels of corn? Nothing. You're just going to slowly die. And that's essentially what happens to microbes when we do exactly that. We're like, yeah, we know that they eat these things and they survive with this much oxygen. We put them in a little plate and then they die because they need each other. They need other microbes to process things before they can eat it. They need other microbes to process their waste products so that it really becomes, this is where it's going to circle back to mining, a true circular economy. And so microbes are the original circular economy, cycling all of the elements on Earth really, really efficiently and completely. And so because we can't culture these microbes and grow them up in a lab, we study them using their DNA. And so my PhD supervisor and I, to this day, and I've been graduated for a while, argue about this, but I think that second to the really horrible picture of evolution where the monkey becomes a human, and that's not how this works at all, and it's caused a lot of angst in the world for absolutely no reason other than it's a poor illustration. Second only to that is this idea that DNA is like a blueprint, because it's not. A blueprint is what's on the screen here. And that is, you can see the shape of the house. You know that it's a house, first of all. It's not an apartment building. It's not a skyscraper. You can see it's a house, probably a pretty big and nice house. The point is, you could also see how big it is, where the windows are, how tall the eaves troughs are, all of these things. And DNA does not work like that. DNA is more like a toolkit. And so when we're looking at the DNA of microbes, we're thinking about it from this perspective, where we're saying, what can these microbes do? What tools do they have at their disposal? And we're looking for really small changes. Did you see it? Did you see the change between these two slides? We're looking for these really subtle changes in these tools. And so here you can see the pickaxe in the middle, the handle size changes size slightly. And we're looking for tiny changes in the DNA that would modify the tools in these small ways so that we could use them in different scenarios. And so when we work with DNA, we're working really in the matrix. And we talked a lot about that. There's a lot of great analogies with the Matrix movie, but we really get these huge files of A, T, C's, and G's, just the 4 letters of DNA that encode for all life on this planet. So we get these massive, massive text files, amounts of data. And we're trying to understand from there, who's there, what are they doing, and who's doing what, how do they respond to change? So these are the fundamental questions that we're always trying to answer. And we do this at Koonkie in a bunch of different industries. So we've done everything from wine, where we're looking at yeast genomes to see, what changes the flavor. What's the difference between wines in BC and wines in Italy, and according to the yeast genomes? Again, going back to that idea of these toolkits. Work in agriculture, work in astrobiology, where we're saying, what can we learn about microbes on extreme places on Earth that then we could use to look for microbes out in space. And so if we know that microbes can live in places on Earth that are most similar to some of the different conditions in outer space, those are kind of good analog environments, we call them, for studying what's here and what we could do there. We also have done big projects on baby poop, adult poop too, but it's less cute, and climate change, and now mining. And so these are the questions, again, that we're asking in all of these scenarios. but we're really at the point here where we want to know who lives in these environments. And I can tell you from some really early data that every single sample that we're processing from mining communities, we are finding new species, sometimes even new genera or families of microbes, but there's a lot of discovery to be done. So just who is there? What are they doing? So what kind of tools do they have? How do their tools change in response to environmental change? Environmental change can be a lot of things. It can be natural climate change as forests develop, as the temperatures on Earth fluctuate, the water fluctuates. It can also be things like mining. So how does adjustment to pH and exposing things to different levels of oxygen and different minerals and breaking it up, how does that change the microbial communities are there? And then what are the consequences of that? So what does that mean for cycling some of these, it could be metals, it can be nutrients, it can be carbon. How does that change the biogeochemical cycles on planet Earth? And then what can we do with that information? So how can we take that information and then start to apply it to problems? So I like this slide. It's almost perfect. If they rounded the corners and then it was a circle, it would be the perfect slide, because then I would be able to definitively say, like, see, microbes are the original circular economy. They have been cycling things over and over and over, millions and millions of years before we were, even remotely a part of this world. And they're going to continue to do it after us. And so what can we learn from microbes in terms of becoming really efficient and developing technologies across industries? And some nice examples of this really are sonar copies bats. And so bats actually can plug their ears when they're screaming so that they don't deafen themselves. And sonar got developed after that. We also see some really cool prosthetics developed after animals that are really fast. And all of this to say, you hear the phrase, like, we don't want to reinvent the wheel again and again and again. And then we bypass biology that has already solved that problem and essentially really do try to reinvent the wheel. And so with microbes, we're trying to say, no, let's figure out how this wheel works here. And then let's apply that. So let's learn from what's already on this planet, and let's learn from what nature can tell us. So this brings us to this really big project, the MMAP, so the Mining Microbiome Analytics Platform. It is absolutely an amazing project. And so mining is at this point where a lot of the chemical and physical solutions are kind of tapped out. This is my understanding. So I'm a total dilettante in mining, to be clear. Microbes, yes. Mining, still learning. But there is, my understanding is that there are these declining grades of ore. And so we need to be able to get minerals and value-added products from lesser quality materials that are harder to get those things out of. And also, from every interaction I've had with mining companies, they really want to be able to do right by the climate and meet their climate goals. And so they're saying, what can we do with microbes to really improve this industry and improve this planet? And we need mining. We have to electrify the grid. That's pretty well established, and we can't do that without mining. And so we're really trying to develop technologies here that are green and reduce environmental threats. And so we're doing that with data. We're doing that with microbial data in the case of MMAP. We're starting with tens of thousands of samples, which is a lot. It's a lot of samples. To put this into perspective, we did a pretty wide search in the public databases of microbial DNA data, and there was just under 450 available samples. If you look at wastewater, there's about a couple thousand. However, most of those are municipal, so it's not really what we're looking for here. So if we're being generous, we could say that there are 700 mining and mining adjacent samples publicly available. And so we're trying to get several orders of magnitude more data from these sites. Why is that important? Because we're discovering so many new species, so many new genes. Everything is about comparisons. So because DNA is not a blueprint, we can't just look at the genomes of one community and know what they're doing. We have to be able to compare them to other known things and to each other in order to figure out what those genes are, who they are, and how similar are these sites to one another. So we need all of this data to really lay that analytical foundation before we can start to ask really good questions and develop really exciting technologies. And so MMAP is kind of doing all of that work. So we have everyone from mining companies, synthetic biology companies, laboratories, software developers, bioinformaticians like my team, to really come together and put all of this together. So there's this positive flywheel effect. And what do I mean? To put this into context, we took a sample from the public repositories from a copper mine, and we ran it through every single taxonomic, so who's there, classifier that is publicly available. On average, we could identify, with this particular method, just under 6% of the community that was there. That's not great, right? So you're looking at 95% of the community, you just don't know who it is. Great. Then we took 6 samples, so only 6 samples, and we pulled out all of the genomes, so the complete set of genetic material for an organism from 84 new species. So with 84 new species, and when we have the whole genome, we can figure out who they are. And we added that back to these tools that we were using that only classified about 6% of the data. And when we added just those 84 genomes from just those 6 samples back into this database, we were actually able to classify about 30% of the community. And so all of this to say, this is why we need all of this data. If we want to start looking at most of our communities, if we want to start understanding most of who's there and what they're doing, we need to have a repository like this where we can have tens of thousands of samples and we can move that from 6% to 30%. I'm not going to say 100% because that's like a pretty audacious goal. But if we could start to get 70 or 80% of our communities known, we can move a lot faster in technological development. And it all really comes down to this data. So we're getting this genomic database of microbes, and it is abstracting all of the bioinformatics work that needs to be done. So what that means is that MMAP will actually take care of all of the work from going from those big text files to a human readable interface that one could interact with, ask questions of, search, do analyses within the platform, so that we can enable things like new bioremediation processes, low footprint resource extraction of low-grade minerals, and it's really this additional layer of information. So we know that these technologies are promising. There's some early work actually out of UBC that shows that there might even be microbial fingerprints for what's deep below the surface. And so imagine you can go and take a sample that's shallow enough to be dug with a shovel, still hard work with the shovel, but could be done with the shovel, and send it to a lab for about $100, process it within the MMAP platform, and get a probability score of how likely there is to be something of value beneath the surface. That is low impact, it's affordable, and it's pretty fast. And so technologies like that are not yet here, but they could be. And so these are the types of things that we're looking to develop, in addition to some of these other things on the slide. So what is the suitability? How can we use microbes to monitor the remediation of legacy sites or improve the remediation of legacy sites? And then this idea of novel gene and sets of genes, gene cassettes for enzyme discovery and pathway engineering. And so synthetic biology can be a really powerful tool, but you can't start from nothing. And so we're really trying to give the whole process this kickstart and say what would normally take people months or years to do, they should be able to do in hours or weeks within the MMAP platform because of the data and because of the tools that are within it. So the mining industry already has these really established concepts of a circular economy. And so again, I'm just putting this figure that I like so much on here again to say, this is really the foundation in my mind for biotechnologies improving this circular economy and really becoming a natural part, hopefully in the future, of every stage in the mining life cycle. So really great way to catalog things that are problematic, for example. So if one mining site is having a problem with a particular bug, it's very likely that that's not an isolated incident. We'll be able to do things like that are more flexible. We know that these things can work. So there's already metal-eating bacteria that can go pretty quickly. And they can eat nails like within days. And then we can do this biohydrometallurgy processes. But we need to have this georeference framework for the basic and applied research. And that's really what the MMAP platform is all about. I'll just leave you with this final thought that I really like, and it ties back into the space theme at the beginning. My contact information is here. I'm always happy to talk about microbes, DNA, bioinformatics. But Stephen Hawking said 5 years ago that we need to find a new planet within 100 years. And whether or not you believe that to be true, and I don't, because I think there's absolutely no chance of that, it is true that we have not yet found collective and sustainable ways to maintain our own planet. And I think that we can look to microbes to start to develop collective and sustainable ways, learn more about each other and how we can really bring each other closer to one another and our home planet. So thank you very much.
Thank you very much, Aria. And thank you also for the work you're doing for the MMAP project. The bioinformatics that you're doing is you know, adds something different compared to the traditional microbiology that you referred to. I'd like to welcome next Professor Sue Baldwin. Sue is a professor in chemical and biological engineering at the University of British Columbia, and she's the co-lead of UBC's contribution to the MMAP consortium. So I'll ask Sue to talk about some of the applications that she's seeing from the big genomic library that we're creating.
Great. Thanks, John. Thanks very much for the introduction. And yeah, welcome everybody to this webinar. It's a pleasure to be here. Thanks very much for this opportunity to tell you all about the work that we've been doing. And so in my presentation, I want to describe how we've used genomics to develop biologically based solutions for the mining industry. The mine microbiome, as Aria described to you from a high level, includes surveys of the many different types of biomes that you can find on a mine site. And by studying these, we hope to design, implement, and operate sort of nature-inspired solutions with the overarching goal to secure a sustainable supply of strategic metals and minerals for the future low-carbon economy. So I'm first going to illustrate some of the work that has been done to date, and then provide some insights into future applications that we are working on. Okay. Oops, there we go. So mine sites contain these very large landforms that are pretty daunting in scale and seemingly totally devoid of life. And components of the biosphere have been stripped away when these large pits are built to extract the ores, and extensive artificial lakes are created, and it's not imaginable that anything living could exist in these lakes. So due to this incompatibility of mined materials with thriving ecosystems, these wastes need to be contained. Otherwise, the consequences will be catastrophic. And we've seen this with several recent mine tailings impoundment breaches that have happened over the past few years here in Canada, as well as elsewhere in the world. And so when dispersed into the surroundings, these water-saturated, fine-grained materials scour out the natural features. So you can see on the right-hand side, this used to be a forest. And now the tailings that were dispersed from at this particular site have scoured away all of the vegetation, destroyed all of the trees, and then covered everything with this hard-packed impermeable material and potentially risks from metals that might be leached out of it. So in this context, the mining industry is facing major challenges in order to meet the United Nations sustainability goals. And these include improving environmental stewardship, adjusting to climate change, and doing that by adopting carbon negative technologies, addressing water scarcity and equality by implementing less use of less water, or better quality control over the existing water, reuse of water, etc. And all of these things will benefit the mining industry as a whole, enable them to manage their risks better, and have more reliable processes to sustain the supply of essential metals. So if we look closely at the structures that are on mine sites, we'll realize that these largely geochemical environments are not totally devoid of life, especially with respect to microbial activity. And the field of biogeochemistry studies life at mineral microbe interfaces. And we have organisms that we call chemolithotrophs, which literally means that they live off of rocks and chemicals and don't require carbon compounds that are derived from plants. So there are entire communities in these environments. We can't see them, but they're there. And as Aria mentioned to you, we're discovering what they are and what they can do with this MMAP project. And so there's an opportunity to leverage the capabilities of these specialized microbes to develop new solutions that we can implement in the mining industry to decrease the footprint of mining, to better select the ores that we want to process, and to sort of improve the interface between the mine and the environment. So for many years and increasingly, microorganisms have already been used and are being used in the mining industry for water treatment. And so this is done so as to remove any harmful chemical compounds before the water is discharged into the receiving environment. And water that's discharged from mine sites is heavily regulated and needs to meet these requirements. And water treatment can involve tanks, as you can see on the left, or more nature-inspired systems that you can see on the right. And either way, they are becoming more common on mine sites as solutions for solving water quality problems. However, challenges still remain with mine-impacted water treatment, especially with respect to their cost and reliability. And also, not many of these current solutions are capable of recovering the metals from the water. So the metals are taken out of the water, but they end up in the sludge of the bioreactors, and the sludge accumulates on the site. So you still have a risk and a liability to manage. And so we need to improve these water treatment systems. We need to improve their reliability. We need to do it in a more cost-effective way. And we also need to be able to recover some of the metals. So I'll describe 3 different processes that we've studied. So in a first iteration of a nature-based process that looked a little different from a natural grassland, used a waste product from another industry as a slow-release carbonaceous material to support a microbial community. And this treatment system was quite effective. It was removing 30 tons of arsenic per year and 230 tons of zinc per year. However, it reached its end of life. It's unable to operate in perpetuity, but it presented an opportunity to understand the mechanisms that were taking place in this process. And so using genomics, we were able to identify who was there. And based on our knowledge of these taxonomic groups and what they might be doing, we were able to reconstruct the mechanism that was happening inside this wetland. What were the different steps and what organisms were involved in those different steps? And we were able to take this even further, where we collected the DNA from this treatment system, and we took pieces of it and we inserted it into a laboratory bacterium. And then this laboratory bacterium was subjected to a functional test to test if some of these pieces of DNA inferred a trait that allowed them to resist arsenic and grow in an arsenic environment and transform arsenic from one species to another in order to facilitate its removal. And so from this information, from the genomics and also from all of the geochemistry and mineralogy that we performed, we were able to reconstruct the mechanism that was taking place in this successfully operating system. And so now, armed with this knowledge, we're able to take this mechanism and recapitulate it in several newer versions of this treatment system that we can now make operate on more reliable way and for longer periods of time. Now here, we don't always use natural wetlands to do water treatment. There are also tanks that are used for treating water. A lot of it is used on mine sites and there are vast quantities of it. And so sometimes you have to build these big tanks. And I don't think there's a person in this picture here, but this is pretty big. However, these treatment systems, they have an advantage over the more natural treatment systems in that because everything is in a tank, you can control the conditions inside the tank. You can control the pH, the temperature, and you can control the nutrients that are added to the bioreactor. And having this better control, maybe we can manipulate the environment to match the exact organisms that we want to grow in that environment. However, this sometimes doesn't work. And so these treatment systems are prone to operational problems. And those problems are caused by microbes. So not only do we have beneficial microbes, we also have those that contribute to operational problems. And some of these are filamentous species that you can see on the left-hand side, and they cause plugging. And when the reactor gets plugged, and a reactor of this size, tens of thousands of cubic meters in size, needs to be shut down, the contents removed, and restarted. So we use genomics to understand what's going on when these operational problems occur. And we can identify who is there. We can identify what these organisms are doing by taking their DNA, assembling it into genomes, looking for the functional genes. And then we can take a look and see what happens when we have this plugging occurring. And we can see which organisms were prevalent at that time, go back to their genomes, see what they were doing and why they were so prevalent at that time. And we can look at the conditions that they like to grow in, and now go back to the control of the bioreactor and see how we can adjust that in order to not have these microorganisms take over the community and improve the reliability of the bioreactor. The last example I'm going to give you is at a more of a discovery level. So selenium is an element that is of concern at many mines, coal mines, metal mines, and also at coal-fired power stations. It's of concern because once selenium gets into the environment, it bioaccumulates up the food chain. And then it causes developmental defects in fish and birds and other animals. It turns out you can remove selenium using biology because there are bacteria that metabolize selenium and turn it into elemental selenium, which is a really good idea because then you can take that elemental selenium and do something with it. But there are many different ways in which microbes can metabolize selenium, and we know very little about them because this area is very understudied, and there are only a handful of organisms that we know about that can do selenate reduction. So we can run bioreactors and do experiments in the laboratory in order to learn more about what organisms grow in these selenium-rich environments and what are their capabilities and how can we manipulate them, for instance, by feeding them different types of carbon sources. And so this allows us to discover very many more microorganisms that have this trait, have the ability to metabolize selenium as a specialization, not because of versatility in their enzymes. And so now we can find out, how can we get these organisms, how can we culture them, and how can we use them? So I've just given you a few examples of what we've done to date, but there are many more things that we can do based on nature-inspired microbial processes. Overall, we want to pivot away from the use of chemical reagents. There are many biopolymers produced by microorganisms that can chelate metals. We want to turn the concept of waste into a recovery of value. For instance, e-waste. We can recover metals from e-waste by leaching it using the organic acids produced by microorganisms, and overall just gravitate towards a more ecosystem-based climate change adaptations of increasing biodiversity and conservation approach to the industry. So thanks to everybody who contributed to this work, all of the industrial collaborators, all the funding agencies, and thanks very much to the audience for listening. Thank you.
Thank you, Sue, and perfectly on time. And I should mention that Sue's original research into selenium and discoveries in the Elk Valley led to Tech's solution for bioreactors. So Sue's been working on this for a long, long time and really is one of the world leaders in this field. So always very exciting to hear Sue talk. I'd like to turn now to Bill Burton. The MMAP consortium is a very big consortium of companies that are really working at the cutting edge of biotech solutions for mining. And one of our long-term partners is BGC Engineering. You would have thought, you know, what's an engineering company got to do with biotechnology? Well, BGC is a very innovative company, and Bill's been a long-term supporter of this work for many years. So over to you, Bill, to talk about your applications.
Thank you, John. Get my screen going here. All right. Assuming I don't see any protest means you're probably seeing my screen. Thank you. Hello to everyone. Thank you for having me. Bonjour à tous. Merci de me recevoir. This presentation I'm going to give, talking about mine waste management and options for biotechnology, was prepared by some colleagues, Julian McGreevy, Wade Gann, and myself. I will be bringing a perspective of a geotechnical engineer, one who specializes in managing tailings, and I'll also be bringing my perspective of working on R&D at BGC Engineering. By way of background, BGC is an applied earth science consulting company. We bring consulting services and software solutions, and our purpose is pioneering responsible solutions to complex earth science challenges. That maybe gives you a hint of why I'm here. We work with mining, pipelines, transportation, communities, renewable energy. That background in mining really has shown us that these big challenges, they need multidisciplinary approaches. Some of these challenges include managing tailings, you know, the silt and sand particles left over after mining processing. And Sue and Aria did good examples of showing those. And, you know, millions of tons of these materials are generated every year as society develops the resources it needs. These facilities can pose safety, environmental risks that must be managed. They can generate contaminated water that must be managed. Makes it all very complex to transition these large landforms to something else, a long-term use at the end that's acceptable to communities around them. Part of the challenge is because both chemical and physical stability must be maintained, and current technologies can be challenged by this. There are times we want waste to be submerged to shut down deleterious chemical reactions. For physical stability, we know that unsaturated soils are typically stronger than saturated soils. So the different conditions are in opposition and there's a tension. As well, when we go to real world conditions, There's all these processes that are going on around these facilities. There's precipitation, there's runoff, there's evaporation, there's chemical processes, exothermic, down at depth. And they're where we can see it on surface, and they're happening below the surface. And mapping out, understanding, and engineering these processes is BGC's role in this multidisciplinary team. So I believe that biotechnologies can play a role in these challenges. For example, what if we could harness some natural microbial processes? Sue's already given us examples. What about the ones we see operating in coral reefs where cement is grown, heavily influenced by microbial activities, or in our teeth for that matter? Could we get to biocementation, to microbially induced cement? Could we use that to strengthen tailings and waste rock? Could we use that cement to mitigate seepage? Microbial processes will often generate gas. Can we use that gas to desaturate tailings? People are studying this. Bioreactors to treat contact water. Sue just showed us those. Bioleaching, biofilms to recover or reduce contaminant generation. I've highlighted biocementation and tailings here because that's what I'm going to talk about the most, but we do see opportunities in all of these processes here. So research, believe it or not, research in using microbes to amend the properties of soil is present and increasing in geotechnical engineering. This year, the American Society of Civil Engineers awarded one of its most prestigious awards, the Terzaghi Lecture, to Dr. Ed Kavazanjian for his work on biomediated solutions in geotechnical engineering. He talks about a few different processes in here. The lecture is available on YouTube. I do recommend it if people want to see this side of these technologies. In his lecture, he talks about MICP. What is MICP? Microbially induced carbonate precipitation. It's quite well studied. It's been in the geotechnical literature for 15 years or so. This work is generally focused on improving the conditions of natural soils, being studied to resist liquefaction from earthquakes and generally behave better under civil structures. The scale of the research work ranges from beakers to small site studies. I'll describe it, and then, but as you think about this, and as I show a bit about it, I want you to think that a lot of this research and technology development has been done in an environment where the tools, leading edge tools that Aria was talking about, Sue was talking about, were still in development. So even though it's been around and has some basis, there's work to be done here. So with MICP, the trick is we want to do carbonate precipitation. And if we can do this with microbial activity, we can create a bond between soil grains. And we can create calcium carbonate through the breakdown of urea. And if we've got the right microbes and some calcium, we can have calcium carbonate come out the other side. Aria said that she was microbes, yes, and mining still learning, and I'm definitely mining, yes, and microbes still learning. So I won't get tons into the technology, but I'll start talking about places where we think this technology can be applied. So what's the goal? The goal is to bond tailing, sand, and silt particles together to increase strength, making it safer and less costly to contain these wastes, easier to reclaim, easier to place covers. If we can make tailings less permeable, if we can fill pore spaces, we can reduce the formation and we can control where contact water goes. We can improve chemical stability. We can coat reactive minerals. We can reduce that water and airflow that drives the reactions, and maybe the cement have some buffering capabilities. But the question is, can we go from the laboratory to an economic deployment in the field? You know, the top left photo here, we see bacteria working around a calcite crystal. On the right is a scanning electron microscope of a grain of tailings with calcite crystals being formed on it. This is some work that my colleague Nicole Woodcock did at Queen's. And so can we go from that scale to the big world scale? You know, tailings facilities are complex. They have different areas. Some of the areas are wet, some are dry. Some of the tailings are coarse, they're sand size, some are fine. And there's all these weather influences going on around them in those processes I mentioned. So field scale application will depend on local conditions. In this sketch here, this schematic, I've got a conceptual tailings facility. There's an engineered structure, a tailings dam around the edges that control and contain the waste. There's these tailing solids that are loose, wet, possibly might flow if released by these structures. There's ponds of water on top. So characterization will be required. Aria's discussion about MMAP is the perfect tool for this, and it's opening new doors, because if cementing microbes, well, they may already be present, or maybe they're not present and we have to introduce populations, but we don't know yet in most cases. And you know, fluids with microbes and nutrients can be applied to simulate these populations in critical areas. We can start to engineer. The little cartoons down here show what we're thinking about, where we've got bacteria working away around a cell, creating the calcium carbonate or other materials, I'll get into that, that join these grains together and fundamentally change their physical properties. I'm going to show a couple of concepts. They're aspirational. This is what we're working towards, and I'll say actively working towards. The first concept is an idea of surface treatment. Can we make a cemented crust on the top? It'll be tricky. We'll have to honor that some parts of the beach, what we call the tailings deposits, are below air, some are below water, so there's variable conditions. But if we could get to a cemented cover, it's going to increase the performance and reduce the cost of reclamation. It's going to reduce infiltration, seepage. It's going to give us new capping options. This figure on the right is from a paper that Gord McKenna and I and some colleagues of ours did a little while back. And I'm putting it here to show that we do not need to have like construction grade cement produced to have measurable and tangible benefits. What this diagram shows from left to right is increasing shear strength of tailings and the different options that come from that. On the left, when your shear strength is less than 5 kPa, you're in a boat or you're in a hovercraft to traffic, and you can only move things hydraulically on top. If you get above 5 kPa, maybe you can't quite walk on it yet, but you can float geogrid and other materials and start to very carefully and in a very costly way place material on top of it. If you can get above 25, if you get above 50, now you're running around and able to use regular construction equipment. These are rules of thumbs on these shear strengths. But the point here is that 50 kPa, when we talk about strength of cement in industrial applications, we talk MPa. So we talk strengths that are, 1,000 times what we're talking about here. This next concept, this is a bit more aspirational. Can we get cementation within the tailings deposits? If we can do that, maybe we don't need these containment dams. But work to be done. I think, although there's all this MICP research and other forms of biocementation, it shows great results. We've got to do work, got to keep doing the research to get it economical at a larger scale. Here's some thoughts I have on things we need to work on. One is enhancing these MICP processes. The microbial processes, as we know them now, with the bacteria we know now, they're slow and they're complex. So can we enhance them? Can we make them go faster? Can we come up with better ways to apply these materials? Carbon sequestration deserves research. There can be CO2 inside these cements. we can get a double bang here for our work. We should be investigating, and people are investigating other metabolic pathways. Can we replace urea? Iron, phosphorus, salt, all can be precipitates. The diagram on the right is work showing sands of grains being cemented by iron. And then we've got to get to these large scale field trials. We've got to test what we don't know. The in situ applications, the resources to do this are a lot more, but we've got to go there, find out the controls of temperature and precip. We've got to develop construction methodologies. We got to develop construction quality processes so that we could do this in a repeatable, sustainable, economical way. I'll leave you with a closing thought of can we evolve the state of practice? Can mine waste containment shift to becoming landform engineering with an opportunity to enhance biodiversity? I am really looking forward to working with the folks at BRIMM and MICA and colleagues at MMAP and everyone on this call to find out. Thank you very much for your attention.
Thank you very much, Bill. And I think you're getting a sense of how big this consortium is. You know, if you really want to tackle a big scientific, technical problem, you need different angles to tackle it. And you know, certainly the geotech angle and putting these solutions into the field is a key part. We don't have presentations here from our two main mining partners, Teck and Rio Tinto, but we certainly do very much you know, collaborate, and we really appreciate the support that they give for this project. They're active partners, and we're taking samples from their mines to direct or create solutions to things that they're interested in. And finally, I'd like to introduce our last speaker, another member of the MMAP consortium is the industrial biotech company Allonnia, based in Boston, but also with Canadian headquarters in Calgary. And Bryne, I'd like you to talk about your perspective on the future of biotechnology in mining. Thank you.
Thank you, John, for the intro. I'm hoping the same as Bill, that I am able to share the screen with everybody and it comes up appropriately. I'd also like to, it's good. I'd also like to thank Aria, Sue, and Bill for your excellent presentations. And I'm hoping that I can actually have a presentation as enlightening and as encouraging as what the three other individuals have presented. And welcome to everybody that is on the call. And I'll start walking through the presentation. And as John mentioned, I mean, the focus of this one will be on biotechnology, future possibilities in mining. And just a little bit on Allonnia. As you can see, a bioingenuity company, and we're dedicated to solving the world's toughest challenges through biology. We at Allonnia believe that solutions to these are found in the world's smallest organisms. And if you go back to a lot of these presentations, they do layer on top of each other, and go back to Aria's presentation, really talked about the size and magnitude of microbes. And we believe that there is an opportunity for the use of biology. In nature, nothing is wasted. Nature is original innovator, and actually recycling everything is a circular ecosystem. And so our ethos at Allonnia is to work in harmony with what nature has already shown us that is capable of doing. And we are wanting to combine biology and technology to fast forward time and what nature brings for future solutions to the present day. Our mission, leverage the power of biotechnology and engineered systems to create transformative solutions for a waste and pollution-free world. A little bit about the merging of factors that have occurred over the last century, and the realization of where biology has come from where it was as early as the 1914, where in one of Sue's presentation talked a little bit about the treatment of wastewaters. That started back as early as 1914, where there was methods of bioremediation where activated sludges were used for treatment of wastewaters. As we go through the timeline, you can see that microorganisms were started to use for bioremediation of oil spills in 1968. 1989, its bioremediation supported a major role in Exxon Valdez cleanup. And these are all combined together of using Mother Nature. And then synthetic biology itself has improved over over that same period of time as well, where there is a breakthrough in the merging of the biological understanding with the powerful technology, especially the computational power, such that in the 2020s, reading, writing, and editing DNA, it's a fully automated system, and it has surpassed Moore's law in the advancements, and it's opening the field to application areas. As earlier talked about, there was, Sue talked about in her presentation as well, that the existing mechanical and chemical solutions are starting to reach their limits. And we need to find alternate methods of improving those, as well as how can biology play a role in those. And when you take a look at the things that have occurred, the urbanization, 70% of the world's population concentrated in cities by 2050. GDP growth, emerging markets, increasing the total waste globally. Sustainable inputs, talked about that in some of the earlier presentations as well, that raw materials are being depleted and are too expensive, and industry is demanding sustainable alternatives. Regulatory focus, there's governments are imposing much more stringent regulations on all industries, and not just mining, but all industries. And one of the earlier comments within the chat spoke about some of those very stringent regulations when it comes to selenium in the waters. Public health, insufficient or harmful waste treatment, directly correlated to multiple health crisis. And ESG, environmental, social, and governance. There is heightened organizational awareness of the goal setting to enhance overall ESG performance. And at a level within the corporations, they need to understand that existing solutions are inadequate and they do not meet the challenges that are in front of us, and that something else needs to be brought forth to help support that. Allonnia, we're focused on 4 different areas that we can see definitely an opportunity for the use of biology, mining, energy industry, industry itself, which is and plastic upcycling as well. Specific to this presentation, though, very much focused on mining. Some of the opportunities for the use of biotechnology that Allonnia sees is impurity removal in mining assets. We see CO2 biomineralization, and this a little bit layers onto the earlier presentation from Bill at BGC, and then rare earth element upcycling as well. To get a little bit more into the details on that of why they're so important, just some mining fast facts. You can see the large revenue base that the profitable industry within mining. There's additional extraction, purification of specific metals and elements, copper, iron ore, coal, etc. Mining products, essential they are to the global infrastructure as well as for a clean energy, and numerous synthetic biology opportunities to improve the recovery yields and lower costs and remediation of contaminants. And the global industry, ore grades are declining. One of the earlier questions as well talked about rare earth elements, but ore grades are declining, and the easy ores they're not as prominent as they were in previous decades, and there needs to be new ways to process the tailings to identify opportunities, and with increasing costs need to be able to minimize capital investments at these sites. Talked about stakeholders demanding better ESG performance as well. High profile environmental issues, disasters. On Aria's slide showed the Mount Poly incident that happened in 2014. Decarbonization, the mining is responsible for 4 to 7% greenhouse gases, GHGs. Water stress due to climate change as well. And in Sue's presentation, talked a little bit about the issue within mining and selenium removal from some of the waters as well. And just as overall, the demand for green technologies, minerals is increasing. And when you take a look at electric vehicles, batteries, These create significant demand for such elements as and minerals such as cobalt, lithium, and nickel. The Allonnia opportunity. So where we really see this is within the rare earth element upcycling. Biology can bind with specific metals of interest, enabling upcycling. The impurity removal for mining assets, again, the use of biology can extract specific high-value ores from complicated mixtures as well. CO2 biomineralization, and this tails off of Bill's discussion as well, that biology can bind CO2 and enable permanent storage in geological structures. Biology, I mean, it can offer significant advantages in the performance and in the environmental impact. It can, reduction of harmful chemicals, as I talked about earlier, chemical and mechanical are coming to an area where their abilities cannot meet the existing needs. Reduction of the carbon footprint, and deployment can be done in situ, in indoor structures, or in contained areas as well. Just to come to a closing on the presentation, one of the examples of impurity removal in assets is that we're presently working with a confidential customer in a sponsored project for removing impurity in mining assets, and this is going to improve their environmental footprint and increase their asset value. So the target was to improve the asset value by 10% and overall reduction of the scope 1, 2, and 3 emissions. From the work that we started back in 2021, there was a large library of organisms that we took a look at and evaluated. Out of that, 20 of the top 20 organisms were selected for optimization. Through that whole process, we continue to take a look at a go, no-go decision making, where we're trying to ensure that the continued work we're doing meets the needs of the customers and also has an opportunity for success. We established the best natural organisms, being the 20, for performance. Continuous testing in representative environmental conditions. The initial work all is based on a lab-based work to be conducted. Testing in representative environmental conditions to ensure that those top selected organisms have the capability to work in difficult conditions, whether it be temperature related, pH related, other competing elements that may impact the success of those, the survival of those microbes. I mean, Aria talked in her earlier discussion and presentation as well, is that microbes for them to be beneficial, need to survive. And if they're not have the appropriate nutrients or competing microbes, they may not be able to survive in that environment. Going into 2022, the next steps is take an advanced biological development, is take a look at the strains, potential development of, enhanced development of those strains, whether it be through evolution or whether it be through genetic modification potentially. And then field testing and deployment is a deployment scenario where you can do it in situ or ex situ, and it's based on the kinetics and the performance deployment at a mining site. And just to provide a little additional insight is that there's several very successful and qualified companies that provide SynBio platforms, and there are several companies that can qualify from a bioremediation specialist. The difference with Allonnia is that we combine all that together, is that we're bringing both the SynBio platforms and the bioremediation specialists, we're bringing that all together and we're combining it so that we're bringing an entire platform of technologies and specialist capabilities to the mining applications, to the mine sites. Thank you very much for your time, and thank you for listening.
Thank you very much, Bryne. And always, I think a feature of the MMAP project as we work on it is the parallel development of the genomic library and also the applications. It's the applications that we're using to test the power of the library, and that sort of feeds back into the development of the library. So it's a very, very large and exciting project. We'll move into some questions now, but I'll just start with a comment, and it relates to some of the technological conservatism that the mining industry has been accused of. And I remember when I first started meeting my parents-in-law, you know, a while ago now, but I realized fairly quickly that they didn't have a dishwasher. And, you know, in the early days of meeting your parents-in-law, you're polite, you don't say anything. But later on, I sort of got a bit frustrated. I said, why don't you have a dishwasher? And they said to me, look, yeah, we tried the dishwasher and it didn't work. Oh, and how long ago was that? Oh, that was in about 1977. And this was well into the 1990s. And I think sometimes, you know, we write technologies off at their very early stage of development, and then we don't look at them again. And what we know is that technological development isn't a straight line. It never is. And often it takes complementary technologies to really make the focus technology work. And this is what's happening in biotech. So we've got computing power, we've got automated genomic sequencing, we can apply big data because we can get samples like we never have before. And this has really changed the game in biotech. So if you've been around in mining for a while and you've written it off like my parents-in-law did, have another look, because this is going to be a big piece of our armory in greening the mining industry over the next few decades. So please have another look. Okay, we might move to some questions. If you would like to put some questions in the Q&A, and I'll field them, and then we've got about 20 minutes, so we've got the time. I'll start with a question from John Clark, and it's not a specific question, so I'll hand it over to the panel. But John says the biggest resource deposits are municipal garbage dumps, which need to be mined to achieve circular economy to reduce GHGs. How can microbes assist with this? We should have Maria Holoshko on urban mining, but would anyone like to tackle this one? It's a very interesting question, and it's actually happening. We're doing e-waste mining in particular.
I could start, John, if that's okay.
Yeah, indeed.
I think we need to basically go towards zero waste. So stop adding things to the garbage dumps. And I mean, already we're doing some recycling of our glass bottles and plastic bottles and paper. I'm not exactly sure how effective that is, but we definitely want to have zero waste, so that everything that you use does not end up mixed together with so many different types of things in this garbage dump. So that's where we should go to as rapidly as possible. That's my answer.
Thank you, Sue. And I see Bryne's come off mute. Bryne, would you like to add a comment? That was possibly from. Oh yeah, go ahead.
Yeah, sorry, John. Yeah, I think just to duplicate off of what Sue said, that I think we really have to take a look at the various different challenges that are in front of us, and actually identify those very specific approaches to how we're going to manage that specific challenge. So I think really just a duplication of what Sue had said.
Thank you. And I mentioned a side project we have off the MMAP project, which is mainly focused on in-ground mining and remediation of mine sites. But we do have a side project which is on mining of e-waste. So if you follow the e-waste story, there's thousands of tons of critical metals that just get shipped to Africa and ended up in places like Lagos and Nigeria to be melted down. The whole e-waste story is a modern tragedy, particularly in terms of how we waste metals. So being able to use biotech to digest these waste materials and then re-extract, particularly the critical elements, that will become extremely important.
I think you're muted, John.
We don't have any questions at the moment, but I just thought I'd like to ask another question to the panel. And one is, at the moment, we're seeing a lot of concerns about critical metals, particularly strategically. There's huge investments by governments to shore up their critical metal supply chain. We're talking about things like the rare earths, tellurium, scandium. There's a lot of these minor metals. But just a question, how specific can the extraction be for some of these metals, with microbes? Can we make microbes that are quite fussy about the metals they like to eat?
I'll start. I mean, yes, we can make things really fussy, which has some advantages because that can really focus on the types of metals that you're going, or even the form in which those metals are. But generally, fussy in one way can be fussy in a lot of ways, which could mean that they often have really strict limitations on then where and under what conditions they'll grow, which can be difficult to maintain as well. And so it's this balance of, yes, you want your target to be really specific, but you also want the rest of the genome to be fairly flexible such that it can live, outside of extremely narrow conditions, as it'll have a broader applicability.
Thanks, Aria. Bryne, I see you, and Sue too. Maybe Bryne first, then Sue.
Yeah, and I think just to sort of add on to that is that when you're taking a look at selective recovery of certain REs, you also will need to take a look at, of course, the economics of potentially multiple bioreactor type structures that may be required for that selected type of recovery, and or at the same time you'll have to do a techno-economic analysis to determine, does it make sense to divide them all up to a very, or combine them into a, maybe 3 or 4 elements that might be worth combining as well. So I think it'll work backwards, that economic analysis that will determine if selective recovery really plays a key role in it, and if it can, there's advantages to it versus, completing it versus not.
Thanks, Bryne. And Sue, did you have a comment?
Yeah, so people have definitely been looking at how to selectively remove metals. And so, for instance, microorganisms can respond to copper. So if you add copper to their environment, they respond to it by producing these polymers that are chelating compounds that can chelate the copper. And so you don't really need to use the entire microorganism. You could actually just have the microorganism as a factory to produce these polymers. And then you can use those polymers in a treatment system. And you can base your treatment system off of solubility, for instance, because a lot of the metals, if you're precipitating them out as metal sulfides, for example, I'm thinking of processes that people have thought about of precipitating out different metal sulfides. They come out at different pHs. So you could have a pH gradient and take out one metal at a time. But yeah, it does need a lot of creativity to think about how to do it.
And thanks, Sue. And we just had a question from Paola in the chat. Are we working with any universities in South America on this? Not yet, but we've had a few conversations with mining research collaborations in South America, particularly in Chile. So, and I think, Sue, your new postdoc comes from Brazil, doesn't it? Is that right?
Yes, I work with a lot of people from Brazil. I've had several people from universities in Brazil and with common interests. So yeah, very good collaborations with Brazil. And also we've reached out to some institutions in Chile as well.
Yeah. So obviously with the amount of copper that we'll need to mine, a lot of that will be done in South America, and that will produce phenomenal amounts of tailings. So the waste recovery, but particularly tailings stability, will be a big story in South America. I might turn over to another question, and this is one that gets a lot of industry focus, which is, mining is one of the few industries that can use its waste to make an impact on CO2 removal. It potentially, tailings could remove millions or even billions of tons of CO2, which is at the meaningful level that we need to address climate change. How, you know, how much could microbiology, biotechnology actually change that? Because at the moment, there are limited, you know, we know nickel does a pretty good job, nickel tailings, but it's not the majority of tailings that actually respond to CO2. How much could microbiology really help here? Are we looking at a big number or a small number? What's the size of the prize, if I can use that term?
I don't have any numbers readily available, but you know, last week we heard from at Genome BC's conference seminar, we heard from Rasmus from Lansden Technologies, and they had some pretty impressive numbers on their ability to carbon capture from microbes. So I think the potential really is high, and it can be done. You know, and he talked a lot about it took a decade for them to scale that technology up, but okay, you know, we've already committed to enough effects of climate change and consequences of those changes, and so if it takes a decade, like, let's get started.
Indeed.
Yes,
Bill, oh, sorry.
Bill first. Go to Bill.
Yeah, it's a good question, John. One I'm trying to work on. You know, the chemical processes that you're describing, the nickel tailings, the ophiolite rocks, the research that Columbia University in Oman and Greg Dippel's work at UBC looking at geologic storage and these chemical reactions. I mean, if you compare those reactions to the amount of these rocks in the ground, even just in BC, you're in the gigaton of sequestration kind of scale. How much microbes would make those processes go faster and better, and all of those things, I think, is a work in progress. In terms of how much carbon CO2 could we get into a tailings deposit, you know, there's only so much space in there. Even a more porous waste rock pile is about 20% pore space. So I think the real answers will come from, can we combine these multiple needs of extraction and landform engineering at the end, and environmentally responsible containment and CO2 sequestration, that when you get all of those processes going at the same time, I think that's where the microbiology plays in, is the way I think of it. If we find ways to get more CO2 and more of this precipitation, well, mining folks, you know, regularly move materials at 100,000 tons a day. So it's the right industry to scale up those technologies as they develop.
Thank you, Bill. We have another question in the chat, and I think this is a good one. You know, biotech startups, you know, struggle sometimes, particularly when they're producing products. And you know, I think Aria knows the realities of running a biotech startup. It's not easy sometimes. But mining biotech startups seem a little harder to get off the ground, according to Patrick, and I think he's right. And part of this is the problem of scale, needing large-scale pilot demonstrations, which aren't always available to you know, sort of biotech startups. So what do you think are the best ways to sort of get around this problem? I mean, obviously, you know, collaborations like we're seeing here between startups and established mining companies is one way, but how else could we do this to try and prove at scale to get over that development hurdle that a lot of mining biotechs face?
I think just you know, speaking as so sorry, John, but yeah, speaking from another startup as well. But I think one of the ways is you definitely need that collaboration, that active engagement with a large organization. You know, at a mining site where you have the resources to the samples, the information, to their knowledge, and then it's also things such as I'll give an example. You know, Aria spoke about earlier in part of the presentation as well. MMAP is a great opportunity to use that library of information that's going to be accessible and available to help validate and confirm, you know, as you're going through your research and development. And then it's collaboration with other industry specialists, subject matter experts. Bill spoke about the areas that BGC engineering excels in. It's being able to work with them as a collaborator. It's been able to work with a company such as Aria that with their capabilities at Koonkie. And I think a lot of those collaborations need to extend out beyond just collaborating with an industry partner, such as you talked about Tech and Rio being partners within MMAP. Those partnerships need to extend well beyond those boundaries, and you need to find other individuals that have got the same sort of mission, same sort of statements, same sort of philosophy of what you do as a company, and be able to leverage their expertise as well. And I think that's going to be critical to be able to expand on this.
Thank you, Bryne. Another comment there?
Well, typically venture capitalists work on these like 7-year cycles. And I think that can be really tricky in biotech where it might take a decade. And so I think there's just that expectation setting as well with investors and collaborators that these things don't happen overnight, but they are going to happen. And so it's just about that patience and understanding. And I also think looking for some of the quicker wins. And so, starting with bioreactors that are a bit smaller and then expanding out. But we don't want to kind of leave those technologies underdeveloped simply because it might take 8 or 9 years instead of 6 or 7. So I think there's some like social attitude shifts that are emerging in this area as well.
Thank you very much, Aria. We have a question in the chat from Rachel James. It's specific for Sue to the arsenic case. Rachel, if you'd like to contact with Sue directly on through LinkedIn, I know Sue's a good LinkedIn user. I talk to her a lot on LinkedIn, so maybe that's the best way to do that rather than going into a deep technical question. I'd ask one more question about the genomic library question. That we're co-developing. The analogy that I like to use is the mapping the human genome, which happened a few decades ago now, and that has really accelerated the development of all sorts of disease treatments, because rather than guessing, we can sort of pretty well design treatments based on what we know in the genome. Do you think the impact of MMAP for mining biotech will be similar? Can we cut out a lot of the guesswork and accelerate the design of solutions across the whole mining cycle, really?
Yeah, I think so. I think even the early results are really promising there. Like I said, to take 6 samples and run pipelines together and go from 6% to 30% of data that's annotated is a really, really positive signal that we are moving in that direction. And it was actually just in the past couple of weeks here that we've got a full, complete human genome sequenced, which is really exciting. And I think just also shows that the timing for this type of work is right. We're able to do so much more with sequence data than we were even a decade ago or even 5 years ago, quite honestly. So the time is now, the data's there. And so are a lot of the analytical tools. So yeah, I think this can have a huge impact.
I forget the exact figure, but I think the original cost of the Human Genome Project was in the billions. And
yeah,
now
it was about $1 a base.
So we're now down to about the cost of a nice car, a Tesla. It's about $40,000 to do the same work now.
Yeah, unreal.
Yeah, it's amazing. Yeah, and I think we might get it just on some final comments from the presenters, just wrapping up here. So, what are some of the most exciting things that we're going to see out of this project and in the next 3 or 4 years in biotech? I think that the complementary technology story is true. We're going to see some massive progress, very quickly now. So I might ask Sue first. What do you think? What's your most exciting prospect for the next few years?
Yeah, what I'm most excited about is, recovery of metals from waste. And in my work, I've, worked with a lot of mine impacted water. And I just see that there's zinc in there, there's copper in there, there's cobalt in there, and selenium in there, all kinds of things, metals are in there. And then I read about batteries, and I read that the batteries, the new types of batteries that are going to replace the lithium batteries are based on zinc. They're based on cobalt. And I said, we've got them. They're in the water. We just have to get them out. And so that's really what I'm most excited about is, and I think it's within reach. I think we can do it. So that, and the other thing that I'm excited about is, the carbon negative technologies. My understanding from global warming and how to combat global warming is that not only do we need to do everything, every single technology that's out there that can reduce CO2 or capture CO2 needs to be implemented. And we need to implement carbon negative technologies. Otherwise, we just won't reach the temperature goals that we're aiming for.
Yep.
Those are my two dreams.
It's a climate control is a carbon capture story now. We can't just get there with climate carbon reduction. That's for sure. Bill, what do you think of what's the most exciting prospect for you?
Well, I always start with the idea of mining being done, as a temporary use of the land, and at the end we've got a landscape that's acceptable to the communities around, and we create biodiversity. That's my core principle that I hold in all of this. You layer on top of that what Sue just said of the potential to have greenhouse gas capture go into the technologies that create those landforms, and that's what I get really excited about.
And the concept of borrowing the land is something I heard at the First Nations Major Projects Coalition meeting. We borrow the land from the owners, and we should be returning it, for their long-term benefit. Absolutely. Bryne, lots of exciting things, but what's your most exciting prospect?
Yeah, well, Bill just took it from me, but it really is being able to take the land, we use the land, we're consuming something that Mother Nature has given us. And the great idea is be able to turn, after we've consumed that, be able to take and turn it back into what it was prior, whether it be a wetland, whether it be just a structure, whether it be a desert, whether it be a forested area. I think the exciting part about that is being able to be engaged in the opportunity to provide a technology that will enable organizations and industries to be able to turn the land back to its prior form, and then as well, hopefully providing a technology that can make the mining industry a much more viable industry. With chemical and mechanical means being at their close to their end or their limitations, is biology another opportunity to enhance and improve the industry as a whole, from environmental stewardship to social to governments as well. So that would be where I see the excitement from.
Thank you, Bryne. And last comment from the bug lady, who was christened the bug lady at the last CIM mining conference. So Aria has a brand now in mining, even though she says she doesn't know much about it.
Yeah, but that's my new brand. Yeah, bug lady. I'm gonna get a t-shirt. I'm really excited about being able to map the mining microbiome. And so, well, it's not a blueprint, it is a map. And I think we can learn a lot from having that. And it takes so many people to create that map in a detailed way. So I'm really excited to have all the ins and outs of the enzymes and functions and taxa that live in these sites, so that all of these other things can happen.
Thank you, Aria. And I should mention before we close that this project couldn't have happened without the federal government's very innovative innovation program called the Supercluster program. British Columbia hosts the Canadian Digital Supercluster, and this initiative to bring all of us together couldn't have happened without the Supercluster. So that's very important to mention that. Last of all, we are still looking for partners and customers to use the insights from this platform that we're developing, this genomic library. Please reach out and contact us through LinkedIn. We are still in conversations with users, mining companies, and we see this becoming the industry, a really important industry asset. So we're open to conversations. Please do that. Finally, thank you, everyone. Thank you, my colleagues. Look forward to our next meetings and the continuing progress we'll make as we proceed to a joint venture incorporation by the end of the year. So, and thanks to everyone for coming, and thanks for your questions as well. Have a great day, and hope you can join us tomorrow for the next installment. Thank you.
Thanks, everybody. Bye-bye.
Thank you.
Bye.